this was one of the species that really created this whole concept, because when we started learning what Cal Davis was able to do with the species, which frankly was in perilous condition, and it was unclear for a long time whether or not there was a sort of a path to save the species. And I don't want to steal your thunder, but Cal Davis did some great work, and now you can see that it's going to take a really long time to recover them, but there is future and there is hope. And Kristin Aquilino, who manages the program, is one of the key reasons why. She works on the white abalone captive breeding program, which I'm sure you'll tell everybody about, right after 2012, when they started to be able to breed these things in captivity. And if they hadn't done that, the species in the wild, it still is in a really precarious position. But with being able to breed them in captivity, which creates hope that we can soon outplant them into the wild. And it really was the leadership that Cal Davis and Bodega Breeding Lab have shown to do that. Before you hear from her, I wanted to take an opportunity on behalf of NOAA and the Fisheries Service just to say thank you very much for this, and to say that you really are the epitome of what can be done and why you're the Species in Spotlight. I'm going to give you our Species in Spotlight Recovery Hero White Abalone Award. Thank you.
I've been joking with people since I heard about this award, which I'm incredibly honored to receive, that I was hoping for a cape, but this will probably be better in my office. So. Thanks very much, Sam, and thanks to everyone here for coming today. Thanks to everyone who's on the phone calling in. It's really an honor to be here today to talk about something I have great passion for. This work, as Sam mentioned, isn't just something that I have done. We have a huge partnership of people involved in this, who I also like to highlight today. I'm going to start by asking everyone what an abalone is. So, how many of you have actually seen a living abalone? How many of you have eaten an abalone? Only a few. How many of you have dove for an abalone? No one. So if we were in a room in California and I asked the same question, then I get very, very different responses. Everyone has heard of an abalone. Most people have probably eaten one, and a number of people would have dove for one. Abalone to California are a lot like Maine lobster or blue crab to the Chesapeake Bay. It's a really important part of who we are. They're sea snails, so they're somewhat like the snail in your garden. They have a big shell on top. Illustration for the Species in Spotlight campaign. There's lots of stuff that lives on their shell. They have these respiratory pores, these holes on the back of their shell. That's how they respire. They pull water through those holes, and it goes over their gills. But my most favorite part of the abalone is actually not this part. It has a face. So it has eyes right here. It's got a mouth back there, some tentacles, and this really strong muscular foot. That's the part we really like to eat. And I have a few abalone shells to pass around for you. The first one is a red abalone shell. This is from a farm in Goleta, just north of Santa Barbara. This is about market size. You can see those holes on top. The inside, they're really nice interior decorators. They have really nice pearly insides of their shells. The other shell I have here is a white abalone shell. And for anyone who's concerned, I do have the permit with me. This is from California. So white abalone, their shells are a little bit thinner than the red abalone that most people, at least in California, are more familiar with. They're a larger part of the fishery. And they have more raised respiratory pores. That's one of the ways that we can identify them. So I'll pass these around. And we actually have 7 species of abalone in California. The red abalone, like I said, are the one that people are most familiar with because they're one of the most abundant species. The white abalone has a more narrow range. It goes from Point Conception down into Baja, and it's the deepest of all of our California or U.S. species. And they're really important to us. Culturally, they're important to us, economically and ecologically. I put these kind of things on a continuum because they're not totally independent from one another. The first picture here is an image of a white abalone shell that was discovered in a midden on Santa Cruz Island, and it's estimated to be over 7,000 years old. So people have been harvesting these animals for thousands and thousands of years. And we still harvest them today. There's shells and things like jewelry and guitar inlays. Mostly we like to eat their meat. And here's a picture of me diving for red abalone in Fort Ross Cove a few years ago. And coincidentally, the recreational fishery for red abalone actually opens today in Northern California. And that brings a lot of money to coastal California in present day. So it's estimated that fishery is worth about $44 million. Fishery has been threatened in recent times. So in 2011, there was a harmful algal bloom that killed off a lot of the red abalone on the north coast of California. And a couple of years after that, there was actually a partial closure to that fishery where the take couldn't happen until later in the day. And we could only take so many per year. I think they knocked it down by 4 or 5 abalone. At that point, we estimate that there was about a $12 million loss to coastal California because of that reduction in the fishery. This year, the fishery has been reduced by 2 full months, and the take has been reduced again. And so that's going to have a big impact on the state. Economically, also, we have these great abalone farms in California. So this is Doug Bush at the Cultured Abalone Farm, where the chalet passed around came from. It's just north of Santa Barbara in Goleta. It's one of 4 farms that we have in California. They also bring in millions of dollars to the state every year. This value that abalone have currently, both where they are in the wild fisheries, as well as this onshore aquaculture production. The other picture here is of an abalone farm called the Abalone Farm, creatively enough, that's in Cayucos around Morro Bay. That was the first abalone farm in California. And then, in addition to our wild harvest, abalone are really important members of our kelp forests and subtidal ecosystems along the coast of the United States on the West Coast. Important for a number of reasons. One, I like to call them the Zambonis of the seafloor. So they basically graze the subtidal habitat. They maintain a really nice cover of this algae called coralline algae, which is a great place for other animals to settle. They make the area that they live in more diverse. They also compete with things like urchins. We have a challenge going on in Northern California right now where, because of the harmful algal bloom in 2011 that I mentioned, because of sea star wasting disease, and because of a few years of really warm water from the warm water blob and an El Nino event, we actually have this bloom of urchins happening. And it's decimating the kelp forest. And even though our water is cooler again, sea stars are starting to come back, the abalone are not thriving because the urchins are really decimating the habitat and eating this kelp. Some evidence that if abalone populations had been more healthy and hadn't been wiped out in that 2011 harmful algal bloom, that they might be able to compete with some of these urchins and have a healthier kelp forest ecosystem. So they really do modify their habitat, and they're important for the rest of the animals that like to live in this area. I kind of skipped over in their value the commercial fishery, so I'm going to delve into that a little bit now. People started harvesting these animals in the mid-1800s. It was mostly Chinese immigrants. The red abalone are the largest of all of the abalone species in the world, and they made up the greatest proportion of the fishery. Most of the initial harvest was from shore or from boats with big hooks, but Japanese brought diving into this fishery, so that they were able to go deeper. They had the big suits, the giant helmets, and the lead boots. And our diving technology has improved over time. Now we have scuba, we have wetsuits. Over time, people were able to go deeper and deeper, harvest more animals. And I think that this is a big reason that the wild populations are not faring very well in Southern California because of this increase in technology. We were just able to harvest so many. This is a picture, or a postcard from the 1930s in Santa Barbara. This kind of image is really common from the heyday of abalone fishing. Abalone fishing peaked in the 1960s and 70s, so this is from even before that. We took a lot of them, and there was a thought at the time that we couldn't possibly deplete the species because they're broadcast spawners, so they send millions of gametes into the ocean, so they must be reproducing a lot. Now we know that even if they are spewing millions of gametes out every year, not all of those gametes are becoming embryos, not all of those embryos are making it through development and settling back where they need to be in order to replenish the population. So we were really over-harvesting them. The other hit to this industry was disease. So there's a disease in abalone called withering foot syndrome. This animal that's on the left is a really healthy red abalone. Its foot covers up its whole shell. The animal on the right has withering foot syndrome. You can see that its foot doesn't take up its entire shell. It's withered. That's caused by this disease that's a bacterium that affects the esophagus of the abalone. The bacterium is a Rickettsia-like prokaryote, and it's interesting because the disease is temperature dependent. So the animal can have the bacteria and be totally fine, at least as far as we can tell, until the water gets warm. And when it's warm for a number of days, above 18 degrees Celsius or so, then the bacteria proliferate, the animal stops eating, it digests its foot, and it falls off the rock and dies. We first discovered it in the Channel Islands in the 1980s, and it was during an El Nino time. Animals were falling off the rock, it was very warm. Black abalone, which we first discovered it in what is the shallowest of all of our species on the West Coast, and so that's probably why they were most impacted. Steadily moved up the coast. It is now present in all of the abalone that we can find in North America, and so this really was the reason that black abalone became listed as endangered after the white abalone. So I'll come back to this disease in a little bit because it plays a role in the white abalone recovery story. But so far, in terms of wild populations, it's mostly impacted the black abalone. I mentioned that white abalone is the deepest of all of the California species, or all of the West Coast of North America species. It's also apparently the most tasty. So white abalone were definitely targeted for their delicate taste. People said they were super tender. Some people said you didn't have to pound them. They were so tender. I don't know if that's just hyperbole or not, but I've now talked to about a dozen people who have eaten white abalone and have confirmed that that was the most delicious one. And I know people who said they'd go diving and they'd see pinks and reds and greens all over, but they were able to look across the substrate and see these white abalone that are often on emergent substrate rather than down in cracks, because they often like to eat drift kelp in the wild, So they would see these animals and they would pass over all of the other species to go target this white abalone. They definitely fetched the highest price at market, and that really led to their demise. The fishery for them was fairly narrow compared to the other fisheries. It was mostly during the 1970s. And we also fished fewer of them than other species. However, there were probably fewer than the other species to begin with, because we fished about 99% of them in those 10 years or so of the fishery. So we estimate we took about 280 metric tons, and there are probably fewer than 2 metric tons remaining. So we pretty much took them all. And even though there's a few thousand now left in the wild, the ones that are left are so far apart from one another that they're effectively sterile. Abalone are broadcast spawners. They send their eggs and sperms into the water column. So if there's a female abalone over here and then there's a male abalone just a few feet away, the chances of those gametes finding each other out in the ocean are very, very slim. So the animals that are left are unable to reproduce. NOAA got together with a number of people in the late 90s to try to decide what we should do about the species. It was determined that they should be listed as endangered. It wasn't something that they thought was going to recover on their own, even with the closure of the fishery, because of the fact that these animals are so far apart and the density was so low. And they became the first marine invertebrate to be federally listed as endangered in 2001. The idea to recover this species was to try to make more of them in captivity and to outplant the ones that we made in captivity into the wild. So this is again the cultured abalone farm in Goleta. This is again the cultured abalone farm in Goleta. And we knew at the time of listing that there's abalone farms all over the world. So we know how to make lots of abalone. This really shouldn't be that hard. Let's just take those methods, we'll apply them to white abalone. People had bred white abalone in captivity before, and then we'll try to figure out the best way to put them out. And that's actually how things started. So just before they were listed in 2001, a facility in Southern California called the Channel Islands Marine Resource Institute spawned some broodstock that were collected. We collected about 20 wild broodstock and created over 100,000 juveniles. So that was really excellent. That happened right away. People were really hopeful about this. Unfortunately, when those animals were about a year old, about 95% of them died from disease. That disease was withering foot syndrome, the one I mentioned at the beginning. At the time, we definitely knew withering foot syndrome existed. We discovered it in the 80s in the black abalone. We knew it could impact white abalone, but we didn't know to what extent. And there was also nothing we could do about it. So at the time, the person in charge of the program had interest in abalone aquaculture, didn't have the disease expertise necessary to tackle this issue. And so NOAA started to look for places that could figure out, is there a way to actually breed white abalone in captivity without having to worry about this disease? The Marine Laboratory, UC Davis's laboratory in Bodega Bay, California, seemed like the optimal place to do that. We have a lot of expertise there. Gary Chur, who is our director, is an expert in the reproduction and development of invertebrates. Laura Rogers-Bennett, who works for Cal Fish and Wildlife and UC Davis, is an expert in what's going on with the wild population of abalone, particularly in Northern California, and that her science helps manage that fishery. And then, most importantly, at the time especially, was Dr. Jim Moore, who's also joined Cal Fish and Wildlife and UC Davis Bodega Marine Lab. He has been really instrumental in identifying and tracking and learning to treat this disease. So here he is at the microscope because he hates getting his picture taken, so this is the only way I can get his mug on here. And he really has, I think, been the biggest thing that has turned around this program. Now we have healthy animals. He developed an antibiotic bath that we can put the animals in that's filled with oxytetracycline, a really wide-working antibiotic or wide-ranging antibiotic. And so we can put the animals in there, it rids them of this bacteria. Then we can put them in systems where we have treated seawater with UV treatment and filtration, and they can't get sick again, basically. So this is a really big deal. If you don't have healthy animals, you can't breed them. So that's been a really big thing that has increased the success of this program. Beyond that are all of these people. So, like I said, you have to have healthy animals. But then there's an incredible partnership here that's going on that has really helped us increase our captive breeding methods. So here at NOAA, folks from Protected Resources, the Restoration Center, and the sanctuaries have all been involved in this. I've mentioned Kelp, Fish, and Wildlife a number of times. We have all sorts of universities, all of the farms in California, aquaria such as the Aquarium of the Pacific, Abrio Marine Aquarium, the Sea Center. All of us have really worked together to increase the success of this program. And I guess I see my role in this as trying to coordinate all of these partners to make sure that we're utilizing all of these resources that we have to best figure out how to save this species, particularly from the captive breeding standpoint of things. And hopefully we can get the outplanting very soon. Not in this picture, but in some ways I'm in all of this picture in terms of my coordination role. The healthy animals and that coordination, we've really increased our captive breeding success over the past 6 years. So this is the number of captive adult white abalone here on the y-axis. So we started out in 2012 with about 57 animals in captivity, and then our success over time. And this is the number of adults again. So we project by the end of this year we will have 12,000 to 17,000 white abalone in captivity. This is easily more than exists in the wild. This is both really scary for the wild population, that most of our animals are in one small laboratory on the San Andreas Fault. Another reason that partners, you don't want to keep all of your eggs in one basket or all of your endangered snails in one tank. But it's also great, it's a great opportunity to save them. This picture here is actually of an abalone spawning this past March. And these are her eggs coming out of her respiratory pore. This is a tentacle down here. You can actually see the jelly layer around the eggs, which is pretty cool. And I'll come back to her in a minute because she's a very special abalone that spawned. Okay, so we're having success. We're hopefully at the point where we're creating about 10,000 new white abalone a year. What does that mean moving forward? Are we where we need to be to actually save this species? We were in a very scary place early on. It's been 9 years with no captive reproduction at all. It's great that we're having that. But what do we need to do to actually ensure this? I will say that that first successful spawning in 2012 was the closest thing to a miracle I've ever witnessed. And Nature McGinn, who was here at Bodega Marine Lab at the time, can attest to this. So we basically were having females spawn sometimes an order of magnitude fewer eggs than they should, and we rarely even had males spawn, and one male spawned this tiny puff of sperm, and I happened to be there with a pipette to pick it up, and we happened to fertilize some eggs. That's not the way to recover a species, and luckily we're beyond that. So what else do we need to do? And there's three main things in terms of captive breeding that we need to focus on in order to get us to the point where we are reliably producing this much every single year. The first one is post-settlement survival. These animals I mentioned send their eggs and sperm in the water column. They're very small at first. They're very sensitive at first when the embryos are first created. So how can we get them through that early life history stage? We need to figure out how to condition them better reproductively to get them to more reliably spawn every year. And then we have a number of efforts going on to try to increase the genetic integrity of our broodstock. So I'm going to go through each of these things in a little more detail here to talk about what we're doing in the program. But first, I'm going to show you a little bit more about what that life cycle is like, so that way it's clear where these bottlenecks are. So I've mentioned a number of times, they're broadcast spawners. This is a depiction of a male spawning sperm. So the sperm would combine with an egg. So once the egg forms, or once the embryo forms, it sinks to the bottom for about 24 hours, and then a swimming larvae hatches out. And this is what that larvae looks like. It swims around for about a week in the ocean. It has a yolk from its mom during that time, so it doesn't eat anything. At the end of that week, it finds a place to settle. It loses all of its swimming parts. It has to grow a mouth and eat for the first time. It's basically going through a transformation that's a lot like a caterpillar turning into a butterfly. It's going from something that swims and doesn't eat to something that crawls and has to eat for the first time. We expect 95 to 99% mortality during that time. That's a lot, but that's natural. I mean, even the best abalone farms get that much mortality, and it's pretty typical for a broadcast spawner. But if we could increase survivorship even by 1% during that time, that would be a lot of animals. So one of our main foci is getting animals through that stage, that really sensitive metamorphosis, to the point where they're about 3 or 4 months old and their mortality drops off a whole lot. And the farms have helped us a lot. So the farms are very good at doing this, and we have developed some of the methods that they use, such as we have these trough systems that we call our cradles for our new baby abalone. It helps maintain the chemistry of the water. It's an easy way to deliver food to them. We put this turf on there because the first year we did this, we learned that When abalone become about 4 or 5 months old, they're like curious toddlers, and they just crawl out of whatever they're in. Maybe that's an important dispersal time for them. I don't know. But it's an embarrassing phone call to make to NOAA that 5 of the 20 abalone that we have from this year have crawled out of their tank and died in the middle of the night. So the turf has helped that a lot. We don't have our escapees anymore. And so I think a lot of our success has come from improving methods at this point in time, but we can do better. And so I'd like to figure out, are there better diets that we can offer them early on? Is there different water chemistry that we should be using in terms of, right now we're treating our water really heavily. We're filtering down to 0.35 microns at that time. We're blasting this water with UV radiation. That's important to keep this disease out, but abalone are herbivores. I'm sure they have a really complex gut microbiome, and so are we eliminating things from our seawater that might actually be important for them? So we're doing some experiments to see, at what point can we kind of reduce the amount of filtration or reduce the amount of UV irradiance that our seawater is getting. Will that improve their survivorship at all? The other thing we're trying to do is figure out what gets them to tick reproductively in captivity. So one of the great things about white abalone is that they become reproductive when they're just about a year old. A lot of abalone only become reproductive when they're 3 to 5 years old. So this presents a great opportunity to actually study what gets them to become reproductively conditioned, because we have a lot of really tiny animals that are 1 or 2 years old. So this here is the gonad of a male white abalone that's 13 months old. So this kind of yellow part here is sperm. There's another male up there, and then this is a female down here. It's more of a gray color. We haven't yet had success getting animals this age to produce viable larvae, viable larvae that are ready to settle. We have had them produce embryos that have hatched into swimming larvae. They haven't gotten through to settlement. My guess is that there could be something to do with egg quality, where the females maybe aren't mature enough to have really, really robust eggs yet. But that's something that every year we try to spawn some of our younger animals to see if they're really ready or able to produce offspring. But we can do experiments to figure out what gets them going. One of the things I think we've really done a good job of is increasing the quality of their diet. So abalone love kelp. They love kelp so much that they'll stand on the backs of their friends here and reach for a blade of kelp that would otherwise be out of reach. And kelp is kind of like a Twinkie to an abalone. It gives them a lot of calories, maybe not a lot of nutrients. It might be good for their growth, but maybe there's some other things that they need in order to reproduce. So we also culture this species called dulse on site. It's a red algae. You can actually buy this at Whole Foods. It's very proteinaceous. They're sometimes putting dulse in chocolate now, which I like seaweed and I like chocolate. I don't always need those two things together, but you can go try some if you'd like. It's very healthy, and it might be one of the reasons that we've seen a little bit of increase in production. But it can't be all of it, because we're still getting, for the most part, 300 to 600,000 eggs per female. We should be getting 3 to 6 million eggs per female. So what else could it be that triggers them to become reproductive in captivity? One of the things that we're doing to try to, I should step back a little bit. To get them to spawn, we put them in a really romantic solution of hydrogen peroxide. It's like a mild thing that we think triggers some of the pathway that gets them to spawn. So they experience this hydrogen peroxide. They think that Abner and Abigail are spawning too. They better jump on the boat, because if you're a broadcast spawner, you got to go when everyone else does. And that method, I think, is working very well. So when we put animals in this solution, they're giving us all the gametes they have, but that's often not very many. So I think that really the key in terms of figuring out the reproduction conditioning is in the 2 or 3 months before we actually put them in those romantic chemical solutions. What is going on then to get them to invest in their gonads? How can we make them really happy during that time? One of the things we do to assess the reproductive condition is to give them a gonad score. We give them a score from 0 to 3 on this y-axis here. And we can just look at their gonad and visually give them a score. We expect white abalone to only reproduce in the late winter to early spring, based on what we saw in wild populations in the 1970s and what we see in captivity as well. So this is a hypothetical model of what gonad score might look like over a year. So we would expect that in the late winter to early spring, gonad scores would be at their highest. The animals would spawn, those gonad scores would decrease, and at some rate they would increase again prior to the next spawning season. Again, this is completely hypothetical. But we can manipulate things in the laboratory to see if we can change that gonad score trajectory. One of the things we're working on now is manipulating photoperiod, so day length. we actually have places in the lab that are attached to astronomic clocks. The lights are attached to astronomic clocks, so they go on and off the same time the sun rises and sets in Southern California. And all of our broodstock are in that condition. Can we manipulate that at all to see if photoperiod is changing how reproductive they're becoming? So we have other places in the lab where the animals are actually on an accelerated natural photoperiod. Instead of going ahead 1 week in time, as we would, they go ahead 2 weeks in time for every 1 week in actual time, or 2 weeks in photoperiod for every 1 week in actual time. So what that looks like is this, where instead of having one peak where animals know that it's late winter and early spring by photoperiod, they might have 2 peaks. Again, this is a model, so if they're responding to photoperiod, then the animals in the accelerated treatment think it's late winter to early spring twice a year. So we can test this way by putting them in these 2 different photoperiod treatments, whether or not photoperiod is impacting their gonad score, and also whether or not there's the possibility of bringing them up to become reproductive twice a year, which would be really great. We could potentially double our reproduction. These guys, so far in this experiment, it does seem like there's differences in gonad size at different times, or gonad score at different times, but we're not necessarily seeing difference in spawning success. So, and this was our intuition to begin with as well, it's probably a combination of things that really gets them to go. So it's probably not just light, it's not just diet, it's not just temperature, it's probably a combination of all of these. So we're just going to start some more experiments probably this fall, where we're going to start manipulating temperature on top of photoperiod to see if that might increase their reproductive condition. And of the 3 things, the post-settlement survivorship, the reproductive condition, and the genetic integrity of our broodstock, the genetic integrity is another place where we've made a lot of progress in the last few years. So one of the issues that we had in 2012 when we were trying to spawn these animals for the first time in about a decade was that we had very few broodstock to spawn. And in fact, almost all of them were siblings and half-siblings from that initial spawning in 2001. Very few of the initial 20 broodstock that were collected were remaining in captivity. And that's okay to an extent. We know from the geneticists that we work with that if we're only getting down to F1s, F2s, that's not too much of a concern for abalone. But long-term, it is a concern. We want to make sure that whatever animals we're outplanting into the wild represent somewhat the genetic diversity of what was there in the past. Now, there's not a lot left, but we want to enhance that as much as possible. So in October of last year, we actually got a permit to collect more white abalone from the wild. We have collected 5 so far. One of them died due to an abrasion that it received when it was collected. And this is something that was difficult. It was very, very difficult to experience, first of all, because you care a lot about these animals. They're really, really precious. There's so few left in the wild. But it's really important to remember what the ultimate goal is of this project. So we know that white abalone that are left in the wild are quite old. They only live to be about 35 to 40 years old. We haven't seen any significant recruitment in decades. The last time anyone saw a juvenile white abalone in the wild was in the early 2000s, and I think then they only saw about 2 of them. So the ones that are left are really old. Their shells are covered in boring organisms, which means they can be quite fragile. Abalone are hemophiliacs. Their blood doesn't clot, so you cut their foot at all, and they're not going to make it. So there is a big risk with collecting animals from the wild. But if we do it in a way where we make sure that no other white abalone are nearby, those animals are effectively sterile. So I like to use the analogy of the white abalone population is like someone on the table in cardiac arrest. Collecting new animals from the wild, we might kill some of them, just like performing CPR on someone who is in cardiac arrest might break a rib. We're going to probably break a few ribs as we try to restore the species, but our ultimate goal is to restore a population. So this is something we're probably going to have to stomach in some ways. However, there's also ways that we can improve our collection methods. So we're working really closely with the Southwest Fisheries Science Center to try to develop techniques to remove animals from the rock without having to use a spatula or an abalone iron and risk cutting them or breaking their shell. So we've been testing methods with ethanol and other kinds of relaxing, getting them drunk type chemicals that we know doesn't have a huge impact. It doesn't have a long-term impact. It has a very short-term impact on them. Basically makes their foot relax, and then maybe we can go in with an ab iron or a spatula and pop them off more gently and increase the success of bringing them into captivity. But that picture I showed with the graph of the increase in success that we've had, that female releasing eggs was of a newly collected wild female releasing eggs. So this March, we inserted new genes into the white abalone recovery program for the first time in 14 years. So that was a really, really big success. We're super excited about that. And we're hopeful that these new animals that we collected will also continue to help add to the genetic integrity of our captive breeding program. The other aspect of this that will give us more control in terms of genetics is trying to figure out how to cryopreserve sperm. We've tried a few times with white abalone. Abalone generally seem to need very different methods for sperm cryopreservation. A number of people have come up with methods for other species, but they're all very different from one another for whatever reason. So we're trying to hone in on the best method for white abalone. But this will help us, especially because our broodstock are distributed among so many different facilities. We'll be able to either save a spawning attempt where only females spawn and no males spawn, or more particularly make crosses to try to optimize our genetic diversity if we have sperm from different areas or different males. And then the final portion of this that really needs to happen, obviously, is outplanting. And all of our captive breeding methods are kind of for naught unless we can really figure out how to get these animals back out into the wild in the best way. Right now, Fish and Wildlife is doing a lot of pilot outplanting with red abalone. They use a number of different methods, but the main one that they're focusing on are these modified lobster traps with broken up cinder blocks inside. And this provides a really nice habitat for juvenile abalone to hang out in. It's relatively predator-free. We can't keep out octopus, unfortunately, which is one of the main predators of small abalone. But this would be a nice place to come back to sort through, see if the abalone are there. There's other groups that we're working with, like the Bay Foundation, who are thinking about green abalone outplanting. And so we're hoping, and also the folks up in Washington doing pinto abalone restoration work, and they're doing outplanting as well. We're hoping that by combining all of these methods, we can figure out the best way to put white abalone back in the wild. And I think that we're at the point where we can do this. So as I mentioned, 10,000 animals a year maybe aren't quite enough to restore the species. It's very close to being enough. But I do think we need to start getting some of these animals back out in the wild. Some of this is a bit political in that we would like to have that exposure of we're putting some animals back. We've taken some new ones in, so let's replace those with ones we're going to put back out. It probably will be a while before we really can understand whether or not some of these outplanting methods are working well. So with other species, we know that sometimes it takes 2 to 3 years to be able to even see the animals that you outplant. They're so cryptic when they're small that even if you put out hundreds or thousands in one location, you don't actually see animals until a few years later because the ones that survive kind of hide out in crevices and they don't become emergent until later on. So it'll probably be a while before we actually know the best methods for this, but it would be great to start getting some of them back out in the wild. Ideal world, instead of giving you a lecture in this lovely library, I'd actually have you all over to the lab, and I could hold up adorable white abalone for you and tell you the story there.
Important for me and my family. It's been a tradition my dad's been going since he was young, and he taught me how to go as well. We have a lot of holidays based around eating abalone. It really brings the family together, and we're able to connect that way and appreciate getting food from the land and providing for ourselves.
Abalone has been really a huge part of my life. As fisheries, are reduced, we realize how many people really come to the coast to do that. There's less abalone out there, but there's more people taking them each year.
Today, red abalone fishing is carefully regulated to be sustainable. However, historical and commercial overfishing devastated most of California's abalone population.
I witnessed cargo nets filled with abalone being brought in by commercial abalone divers.
The problem that we saw early on was that there were too many abalone being taken.
There would be red abalone and green abalone and pink abalone and white abalone.
White abalone were targeted for their delicate taste.
It was quite tasty, very tender, more tender than the red abalone we have in Northern California.
I found it to be particularly good. They also commanded with white abalone a higher price.
And the white abalone have disappeared. That's a tragic loss for a wonderful craft.
White abalone will likely go extinct very soon if we don't do anything to save them. Right now, the abalone that are left in the wild are so far apart from one another that they can't reproduce successfully. We're breeding these animals in the controlled conditions of the laboratory so that we can bring them closer together for reproduction. and also so they have optimal conditions for survival. We have been incredibly successful so far with our captive breeding program, and we're really excited about the thousands of animals that we now have in our lab. We now have more white abalone in captivity than probably exists in the wild. And as sad as that is, it's also a great opportunity to save them. We are really excited in the next few years to start putting some of these captive bred animals back out into the wild to help save the wild population. There's still a lot of research that we need to do to test how they're going to be able to deal with things like diseases and temperature fluctuations and predators that they are not experiencing in captivity. So we're really hopeful about how they will do once they're in the wild because their habitat is fairly pristine. We, as humans, were really responsible for the decline of the species, and so I really believe that as humans we have a responsibility to help restore them. Ideally, we can recover white abalone populations to the point where they're self-sustaining, and hopefully they can end up on our dinner plates again one day.
Scientists at Bodega Marine Lab are working to ensure all 7 of California's precious abalone species continue to enhance our culture and our economy.
I hope that abalone will be around for generations and generations so I can teach my children how to abalone dive and we can continue a tradition.
If I can hope for one thing, it's that my kids respect the resources that are out there because quickly they can be gone.
So this video was produced by Ben Walker and I, and the target audience is certainly people coming to Bodega Marine Laboratory, people who are abalone fishermen and who connect to the resource in that way. Is special about this program is that we are trying to restore species, but we're doing a number of other things at the same time. So I mentioned a lot throughout this talk of the ways that we're helping hopefully the wild fishery. We're helping CDFW understand the population dynamics of abalone, and maybe that will help manage that fishery better. The other thing that I think we're really contributing to is aquaculture, especially in California, but throughout the U.S. Abalone, like a number of other mollusks, are in danger of things like effects from harmful algal blooms, from diseases, from ocean acidification. And a lot of the work that we're doing at Bodega Marine Laboratory, and with this program in particular, will help our farmers do a better job of maintaining and growing their businesses through the future. So we know there's a huge seafood deficit in this country. We import 90% of the seafood that we eat, and being able to help those growers do a better job and in their operations will improve our global food security. And that's one of the reasons that they're so keen on working with us, because they know that these threats exist. They are worried about the impacts on their business, and we have the resources to tackle them because of this great collaboration that we have with the universities, with the federal and state governments, and with all of our private partners as well. So a few take-homes we have here. Oh, and I would like to eat one day. If everyone's telling me that they're so delicious, it would be kind of nice. And there's precedent for this in Atlantic salmon, having some fishery or aquaculture-based operation for an endangered species. We're clearly not at that point yet, but our wild fisheries are not going to obviously sustain our need for seafood globally, and so perhaps being able to farm this species, if it really is the most tasty and delicious of all of our species, is something that would be a goal in the future. So our take-homes are white abalone are culturally, economically, and ecologically important to this country and around the world. The white abalone will go extinct very soon without our help. We have the tools to save them, and they should be a relatively easy species to save in the sense that this is a numbers game. We're at the point where we're producing 10,000 a year. If we can get those out in the wild and have even a small proportion of them survive, then we should be able to get breeding populations of them in the wild. And that's really, really exciting that this is very doable. And then I think we really need this continued enhanced support for captive breeding and outplanting to allow us to save them faster and more efficiently. So with that, I'd like to put my contact information up here. We are all over social media on Facebook, Instagram, and Twitter. Both I am and White Abalone. And as I've said before, I would really much rather you guys all come to the lab with me because I'd be much more comfortable and I'd be able to show you all sorts of adorable baby white abalone and adult white abalone. So we've had a number of NOAA visitors in the past, if you ever find yourself in Northern California, please let me know. I'd love to show you our lab. Thanks very much.